Okay, class. We are going to start with discussion about this DNA finally. So we hear a lot about DNA these days. Many people say our company has a DNA of innovation, or innovation is our company. I don't have anything in my DNA. Something like that. Okay. So DNA is, I think, that's most like characterized, I would say, most discussed. The molecule, the molecule of the Earth or something. Okay, it's very famous molecule, I would say. So DNA was, I think, it's discovered much earlier, but the structure of DNA was later discovered by the Jim Watson and then Francis Crick. That's the double helical model of the structure of this deoxyribonucleic acid. Okay, abbreviation is DNA. So DNA is the substance of inheritance. It says this is the most celebrated molecule of our time. We have even song about DNA, and then we have about the certain rap about the DNA, as you remember. So what we are thinking is this hereditary information is encoded, coded in DNA. And so, this is kind of like our basic, like, CPU in each single cell. I said DNA is the most important molecule in the cell. Okay, so I can argue that all the time. I think that everybody agrees. So this DNA molecule really, it contains the program and comment, directs the this development, regulation of the biochemical reaction, formation of anatomical structure, and then the physiological layout, and then even it controls the behavioral trait, in some extent, as far as we understand. Okay, so this guy name is Francis Crick. Okay, and Crick, isn't it? Right. Okay, and then not Click, right? And then this guy's name is Jim Watson. I think that he's from England. Okay, and I think he's from Jim Watson is actually from Chicago, Illinois. So they are posing in front of this their double helical DNA model here. I think that's, I can say, DNA is the discovery of DNA, definition of this DNA double helix is through the beginning of the molecular biology that we kind of like we kind of like take advantage of, okay, these days. Okay, so in early 20th century, I think that's in the previous chapters, we talked about late 19th centuries, like Mendel's work, and then Mendel's work was rediscovered by Vauvary's, and then Debris or something, those people, and that lead to the we kind of like that is the chromosome theory, okay, of inheritance. I think that we learned about it. That is what T.H. Morgan, right? Okay, Morgan. Okay, And then I think that's one of the more, advanced we need to add to in 20th century is the microscope. Okay, technology develops, so we have some better tools. From this, really, we look at the chromosomes. Okay, and then we know that chromosome is protein, and then DNA. Largely nucleic acid. Okay. So let me go back. Okay, I'll go to the next one. Okay, so this T.H. Morgan's the word, point to the chromosome, and then the we know that DNA and protein is in the chromosome. So 1 or 2 is the material, genetic material. I heard that the people didn't really pay much attention to DNA. DNA is kind of like a linear structure. People thought that DNA is just a scaffold, and then protein is the one really containing the genetic material. So they have to do some more experiment, and then found that the DNA is the genetic material. So that was done by this Griffith experiment in early 20th century using 2 strains of bacterium. I think that Staphylococcus, Streptococcus, strain that the has the 2 strains. So that is, one is S strain. It has the coat, okay. Strep, I would say strep, right? Okay, and then S strain has they have a coat, and they are really nasty bug. Is a pathogenic bacteria because the coat causes the immune reaction. And then also there's a rough strain. They don't have coat, no coat. Okay, so it's a different type of the same species. It's a mutant, okay? So they are non-pathogenic. Okay, so if you inject this, S strain, okay, to a mouse, then mouse will die because of their sickness. But if you inject the R strain, not dead. Okay, I think that not sick. So Griffiths, Frederick Griffiths used this property, and then tried to show DNA is the genetic material. So what he did is that, okay, he take the pathogenic strain, which is the S strain, and then he killed them with a heat. So they grow them in a okay, so you grow, this is the bacterial soup, and then you put burning flame here, so you boil them, so it is killed. Okay, and then you have the killed substance, isn't it? So which means that maybe spent media or something like that. Okay, so juice here. Okay, boiled, right? And you have that is the let's say S strain dead S strain particles. Okay, right. So and then with the living cell of the harmless strain, so they mixed it. Okay, right. So you have this, and then I will use this, and then you have this have an R strain in it. Okay, so you take this one out, and then you pour onto here, and then you maybe mix it well. Okay. So what they did is that, okay, so you just mix them, make sure that you heat cooled. I mean, you need to cool them in the ice or something, and then you put it there, and then you shake it very well. Okay. And then you use this R strain and then inject into mice. Okay, and then what will happen is that although that was the R strain, but if that was mixed with this, debris of the S strain, which is dead S strain, some mice died. So we'll have the figure here. So this is it. So this is the living S strain, and this is a separate individual experiment. Okay, right. So to demonstrate the virulence of this S strain, you inject them into the mouse, then mouse will die. And then the R strain, rough strain, okay, is a control. You inject them, and then mouse will not die. So what they did is that, okay, this is a heat-killed S strain. So as a control also, heat-killed S strain, you send it to them, you inject into the mouse, they are still healthy there. But what they did is that my previous, picture here, this guy, you mix them, okay? And then you inject them, and then mouse dies. I think that's some died. Okay. So, using the dead mice, okay, they isolate the, bacteria in there, and then they were able to isolate S strain, not the R strain. Okay. So that is what their conclusion is that, okay, something in the dead S strain transformed R strain to S strain. So that is their conclusion. Okay, this is their conclusion. Okay, so that is the what they found. Okay, and I'm gonna move to next one. Okay, and then they I think that a few years later, Oswald, Avery, McCarthy, and then MacLeod, these people announced that the transforming substance, that they saw it in the transforming substance from Griffith's experiment, right, is the DNA. So they use the I think that's in the figure, they treat with a different type of enzyme that kills only protein, kills only DNAs or something. And then they found that, okay, DNA is the one. So their conclusion is the DNA. Okay, but I mean, that is the changing the genetic phenotype, right? So DNA is the material, but I mean, still people somehow did not really buy it because the still skeptical. Mainly, we don't really know much about the DNA, and then also how we don't really know how it looked like. And DNA at that time when they isolated, it's very, very simple molecule. You see some sugar, you see some phosphate, and then you see some weird molecules, but very, very uniform molecule. They didn't really think that DNA will carry so many different information to control the cell physiology, basically. Okay. So later on, this blender experiment, they call it Hirsch and Al Hirsch, okay, and Martha Chase, okay. They did a T2 phage experiment. Another name we call is a blender experiment because they really use the blender, kitchen blender for this kind of experiment. Okay, so using phage. So phage is the virus, bacterial phage. So Al Hirsch is this man. Is Al Hirsch. He is at that time, maybe he's still smoking. This is the picture in 1954, you know, with in the this phage meeting. Okay, I have one more arrow here. His name is Francis Crick, and this guy is the Sydney, you know, Brenner, who started all our entire field that I studied. So, you know, this is phage molecule, you know, I mean, phage, a bacteriophage. As you can see, it's kind of like a you know, lunar module on the cell surface. I think that's your book may say is a lunar module, okay, on the cell surface. To me, that, you know, it's kind of like my dog, okay? But the, you know, as you can see, they landed on the cell surface, okay? And then they are injecting DNA. So this is the DNA molecule. Okay? They are injecting it. It's a very, very nice picture. This is the bacterial cell they are going. So this is the capsid, you know, virion particle. Usually these are composed of protein. And then they have a DNA inside. And then when they infect, DNA goes through and then going into here. And then they can make, you know, more phages, you know, later because of this DNA they are injecting. Okay, so 1952. Okay, Al Hirsch and then Martha Chase performed the experiment showing DNA is the genetic material using this T2 phage. Okay, so to determine this, they designed an experiment showing only one of two components of DNA, T2, which is either DNA or protein, is entering into E. coli. In the cell, you know, E. coli cell during this infection. So which one is going into the DNA, you know, phage? Let's say whatever the molecule they're going into the, you know, cell, then there can be a genetic material. So as you can see here, I mean, we all know already that it's the DNA, but at that time didn't know whether they are going, DNA is going in or protein is going in. So they concluded that injected DNA of phage provides genetic information. This is the blender experiment. Okay, so let's go. So they have two batch of experiment. Okay, so one batch is that, okay, they somehow grew the phage, you know, I mean, somehow labeled the phage with a S35, with a sulfur, radioactive sulfur. So sulfur has, you know, was it protein only has sulfur, sulfur, okay? But DNA is no sulfur. They don't have sulfur, the atom. And the other one is the labeled with a P32, you know, somehow phages. And then how they, the reason why they use P32 is that P is in DNA, but not likely in protein. Okay, so they labeled, you know, so labeled here's a phage. Okay, and then here's also phage. Okay, right, and then they infect those phage into the bacteria here. Okay, then when they go in, infect the surface, and then they will, you know, inject whatever inside, whatever the molecule they have it, they'll put it inside, right? Okay. So, and then when they're growing, then they use the blender because they want to detach the whatever something that attached on the cell surface of the you know, the bacterial cell. So they kind of shake it really hard, then it will be detached. Okay, and then they'll isolate, you know, whatever the pellet. Okay, that is it's a cell, and then they survey the radioactivity, and then in the first experiment, in the cells that isolated in the pellet, there's no radioactivity. But in the second batch, in the isolated cell here, this is the isolated cell, has this radioactivity because the phage DNA is tagged with a P32. Okay, so that is their conclusion. I mean, this is the looks like it's very simple experiment, but this is really hard to do or hard to come up with at that point. And the additional evidence came from that DNA is the genetic material is the Erwin Chargaff. Okay, he see the chemical composition of this nucleotide, polymer, right? I mean, they know that this is the nucleotide, and then it's composed of ATP, GTP, CTP, and TTP, but he did a survey of it and then tried to see the ratio of it. And then what they found is that ratio of A is always same as T. And then ratio of C, which is cytosine, okay, is same as G. So amount of the C is same as G. I don't think they can do really exact same because, chemical method, there's no such thing that you can make exactly same or something, okay? Because we always have some kind of errors, isn't it? Right. So from here, and they found this. And then this ratio is depend on species, okay? So AT and GC ratio is always same in no matter what kind of species. But AT ratio versus GC ratio is really depending on, between species. So that is the base composition of DNA varies between species. But in any species, the number of A and T bases are equal, and the number of G and C bases are equal. This is the what we call Chargaff's rule. I think that Watson and Crick's paper, they quote this Chargaff's rule in their first publication. So basis for this rule was not understood until the discovery of the double helix. So this is the one strand backbone of DNA, and then DNA is composed of this 4 different nucleotide. And you always have a 5' end, and then 3' end is based on this, ribose sugar. Actually, we call it a deoxyribose, isn't it? Okay. So in a sugar backbone is linked by this phosphate, isn't it? So phosphate is the linking. This is the first number 1 nucleotide. And then how the number 1 is adding to number, number 2 is added to number 1 is that, okay, they are using this 5' phosphate is added to the 3' carbon of this, deoxyribose sugar here, right? So they have an OH group here, and then OH group is removed, and then this phosphate group attached to, and this is also, was it covalent bond. So this is how they are, adding the backbone. So this is like we call this a backbone. And then it's composed of this nitrogenous bases. So base is the either thymine, adenine, cytosine, and guanine. These are the one that gives the different, property of each different, nucleotide. I mean, this is what makes the difference between the bases, nucleotide. One more thing, I think these and these are the purine, right? Right, and then I'll just use it. These and these are pyrimidine. Okay, so and then they are going like this. So any kind of DNA molecule, the one end is what we call a 5' end. The 5' phosphate, and then 3' is always OH group. Okay, so always start with a 5' phosphate, and then finish with a 3' OH group in the backbone. Okay, so let me finish here for the first segment of this chapter 16, and I'll continue on next segment. Thank you.